One of the most important roles of proteins in our bodies is as catalysts. Our protein catalysts are called enzymes. So, of course, enzymes, like all other catalysts, increase reaction rates without being used up. The majority of the enzymes that our body uses to control our reaction rates are globular proteins. There are some other biomolecules, such as RNA, that are used to catalyze reactions, but it's primarily proteins that perform this catalytic role. Now, there are a number of different reasons why our protein catalysts are so important in the body. The first reason is that our body needs a very high reaction specificity to avoid negative side effects. So if we look at a typical organic molecule, right, which again, of course, are very similar to the molecules that we have in our body, there are many different reactions that you can have for any given molecule, right? Particularly when you have multiple different functional groups within the same molecule. So like our structure here, we have carboxylic acids, we have an alcohol, we have alkenes over here, we have an ether. There's a lot of different complexity in terms of our functional groups. So you can imagine there's a ton of different reactions that we could have here, right? We could remove our group over here on the right to create some sort of aromatic ring system. We could do some nucleophilic substitution and change what groups we have attached to our ring structure. These are only a few of the different possibilities of how we could react this one structure here. Right? So when we look at our biological molecules, our metabolites within the body, they have many different potential pathways for how we could react them and decompose them or synthesize them into other molecules. So our enzymes, their goal is to make only our desired pathway favorable, right? So ideally, the rates to all of our other possible products will be really slow within our body. And then we'll have a specific enzyme. So in this case, it's this coarse mate mutase enzyme that specifically speeds up our one reaction pathway. So this one goes quickly, so that then our product will always be the blue product here, and we'll only get very small amounts of our other products, right? So it's mostly channeling all of our molecules along this enzyme catalyzed route. Now, another key component to this is that our enzymes also have to be active under much milder conditions than we typically see, again, in organic chemistry, right? So within our cells, as we've been talking about extensively, our pH, right, has to be maintained quite carefully, right? That pH changes and we start having our amino acids and things like that getting ionized and changing their charges and changing the interactions that occur between those amino acids. So it's very important that our pH stays mostly around that pH 7 range. We also, of course, have a specific body temperature. 37 degrees Celsius is the standard body temperature. And if we vary from that body temperature, it will cause huge problems for our body systems overall. Contrast this to what we see in organic chemistry, right? If you're going to react something, usually one of the first things that we do is we heat it up while we're reacting it, right? Heat helps to speed up our reactions. So it's relatively rare that we would do a reaction just on the benchtop at a relatively low temperature, like 37 degrees. So our enzymes then are critical to speed up our reactions that they're occurring on a more useful time frame, right? So we need higher reaction rates to fit in with our biologically useful time frame, right? If you're thinking of escaping from a predator, for example, you need those reactions to be occurring very quickly so that you're producing the energy you need and you're contracting your muscles as you're moving your body. So we can't have these reactions just occur slowly as they want to go along, but we need to be able to speed them up to make them occur when we need them. The other thing that's very important for our enzymes is they allow us a capacity for regulation of all of our biological reactions, right? So we don't just want our reactions occurring whenever. This will lead to buildup of products that we might not actually want, right? We want to be able to produce the product we want quickly when we want it, and then we want to be able to turn it off when we don't want that product. So in the case of our proteins, all we need to do is change the structure of our enzyme here to turn it off, and we can effectively shut off this reaction pathway, right? So our enzymes act as a great mechanism for controlling our reactions, right? You can use things like allosteric regulation, like we saw with our hemoglobin in our previous examples. We can use that to change the interaction between our enzyme and its substrate, the molecule that's binding to and reacting, and then we can affect the reaction rate there and regulate our reaction. Going back to the specificity of our enzyme reactions, it's also very important that our enzymes are highly selective in terms of what substrates they bind, right? So we don't just need to control what reaction happens, but we also need to be very selective about what molecule uses that reaction, right? So there are a lot of different molecules in our body, and some of them will look very similar, right? So an example here is our phenylalanine hydroxylase. So it catalyzes this reaction of phenylalanine, shown here on the left, to our tyrosine, shown here on the right. Now, of course, if you compare this to our other enantiomer of phenylalanine, we need to be able to selectively not bind to our enantiomer, because we don't want to be creating the wrong enantiomer of tyrosine. So in general, we want to be fairly selective about what molecules we're able to bind and react with. Now, it is still true that we can still have multiple molecules that might look similar, binding in the same enzymes and reacting there. And this is often exploited by our drug development, right? So for example, this structure here is actually a drug molecule that's used to inhibit the phenylalanine hydroxylase. So it will bind, but it won't react in the same way, right? We can't add our hydroxy group because it already has it on our ring here. So we are highly selective, but we can still kind of manipulate the selectivity in order to help to regulate our reactions even more. Now, as we've seen in our previous lectures, again, our protein structure is going to be relating directly to what substrate we're binding, and then how we are catalyzing that reaction and speeding up our reaction rates. So our enzyme is going to have a particular binding site that will bind to our substrate and form that enzyme-substrate complex. And of course, we'll have key amino acids that will be forming interactions to that substrate in order to hold it within that binding pocket. So those key active amino acids are marked here in red. Those are going to be binding to our substrate, and then, of course, pushing it to make our reaction. In order to gain a better understanding of how our enzyme affects the rate of our substrate's reaction, it's important that we take a closer look at the thermodynamics of enzyme-catalyzed reactions. So, of course, as you should remember from earlier chemistry courses, enzymes do not affect equilibrium, right? So the equilibrium constant for your reaction will not change. You still will have one product that will be preferred, and the other product or the other reactant that will want to be consumed. So again, you'll have a specific ratio at equilibrium based off of that equilibrium constant. That doesn't change. Now, of course, along with that, our enzyme is also not going to change the free energy of the reaction, right? So free energy relates directly to the equilibrium constant. We have an equation, right, that we can use to calculate between our free energy and our equilibrium constant, right? So if a reaction is non-spontaneous, it's still going to be non-spontaneous when we have an enzyme, right? An enzyme can only change the rate of a reaction. So they will increase reaction rates. So that's our small k, our rate constants. And they do this by decreasing the delta G of our transition state, right? So this is the activation energy for our reaction. It's helpful to look at this from a graphical perspective using a reaction coordinate diagram. So remember, in a reaction coordinate diagram, we normally have our energy on the y-axis. So this is free energy, our delta G. And our reaction coordinate is on the x-axis. This is just showing the general progress of our reaction. So we're starting with our substrate over here, marked S, and we're going to some product over here, marked P. Again, the free energy difference here, the delta G, between our two, our reactant and our product, this is not affected by our enzyme. So you'll note this will remain unchanged. Of course, for every reaction, we have an activation energy barrier as we move into our transition state. So this is that unstable state where we're kind of switching from one form to another. You can think back to different intermediates that we might have talked about in organic chemistry, like carbocations and things like that. You have those generally quite unstable intermediates that will then lead to your final product. So let's look at what happens when we bring in our enzyme. So we have the same diagram here, our free energy and our reaction coordinate versus the showing the change in our substrate over time. And you'll notice for our uncatalyzed reaction, again, we have that free energy barrier, our activation energy. And in the blue here, we have our catalyzed reaction, where it has reduced that activation energy barrier, right? So now we just have these small bumps in between our substrate and our product, rather than this large activation energy barrier. Again, note that this gap here, our free energy of the reaction, is the same. There's no difference between the free energy of our substrate and our product compared to the uncatalyzed reaction. Now, of course, our reaction does get a little bit more complicated here. So you'll notice we have two new symbols here, ES and EP. So we actually have three different steps here, right? Our substrate binds to our enzyme to form the enzyme-substrate complex. So this, of course, is a similar equilibrium to our protein ligand binding equilibrium that we looked at previously. We just have different terms here for, of course, substrate instead of ligand and enzyme instead of just our generic protein. We then have a second step where we actually catalyze our reaction here from the substrate to the product while it's bound to our enzyme. Again, this is where the enzyme is doing its magic to lower that transition state. And then we have a third step where we need to actually release our product, and we would have product at this point, and we would also have free enzyme here. So this is essentially a dissociation reaction. So we would have a KD, a big KD, that would be looking at this dissociation equilibrium. Of course, we have an association step here first, which would have a Ka equilibrium constant for that process here. How effective are our enzymes at increasing reaction rates? Well, if we calculate the changes in our rate constants based on our enzymes, what we see is we see orders of magnitude higher rates for our enzymes compared to the uncatalyzed reactions. So you can see in this table here, we have a list of some of the typical enzymes that we see in our cells. And you can see the reaction rates here in the presence of these enzymes are just astronomically large, right? They're all multiple orders of magnitude faster than what we would have in the absence of our enzymes. So they're extremely effective at making our reactions happen very quickly. And again, as I mentioned, this is very important, right? We need these reactions to happen quickly on our biologically useful time frame. And then, of course, that also allows us to turn off that enzyme and effectively stop that reaction if we don't want any more of that particular product. So both that speed and that regulation is very important here. So how does our enzyme actually lower that activation energy barrier in order to speed up our reaction? So this, of course, is going to relate back to our protein structure that we've been learning about over the last few weeks. So our enzyme is going to use those interactions with our substrate in order to lower the free energy. It's going to do this in a couple different ways. One of the key mechanisms that it's going to use is organizing our reactive groups into close proximity and proper orientation. Right? So one of the things that will slow down an uncatalyzed reaction is that we have to take our two different reactants, right, which are very free to kind of move around and do whatever they want, right? So we'd call that a high entropy state, right? Because they have a lot of freedom, a lot of disorder there. So our initial reactants for an uncatalyzed reaction will generally have high entropy. When we go to form our reaction, we're going to have some sort of transition state where they're both kind of linked together. They might be covalently linked together in that intermediate, or they might just be next to each other reacting, but they're going to be in some specific orientation as they're performing that reaction, right? So they have one single restricted transition state where they're converting between the reactant form to the product form. So again, if you have one restricted structure, that's very low entropy or very high degree of order. So this is entropically unfavorable, right? We're decreasing our entropy, which would be a non-spontaneous process. So that's one of the things that creates that large free energy barrier that we normally see for reactions, right? We have to go from something that's entropically favorable, right, a high entropy state, to a low entropy state in our transition state. Even if you consider a reaction that only involves one molecule, a unimolecular reaction, you still have some restriction of your molecule as you are going through that reaction, right? So you might have one molecule that's kind of flexible and moving around, and it might be reacting between two different reactive sites on that molecule, right? And so it's going to have to bend into one specific shape to bring those reactive sites into close proximity in order to have that reaction. So you're still moving from high entropy to low entropy in an entropically unfavorable process. Compare this to our catalyzed reactions. So in a catalyzed reaction, we are still going to be bringing our substrates into a specific ordered arrangement in the proper orientation and close proximity. But the difference is that we can use our binding energy, the energy of our interaction between the enzyme and the substrate, to help to pay this entropy penalty, right? So again, as always, our delta G is equal to delta H minus T delta S. So of course, our bringing those reactants into proper orientation is entropically unfavorable. That's our delta S term over here. But our binding energy will contribute to a negative delta H in order to help this process be more spontaneous, right? So we can modify our equation essentially to improve our delta G. So our entropy cost is paid using a delta H of forming that positive interaction. I should mention there could also be some delta S components there as well if you have like a hydrophobic interaction between your enzyme and your substrate. But in general, you have some entropy penalty for bringing those reactants into a specific orientation that's essentially getting paid by the enzyme binding to the substrate or the transition state. So your rigid reactant complex then has a transition state conversion that's closer to entropically neutral, right? Because you already have bound your substrates and put it into that proper orientation using your binding energy. And now you don't really have to change a whole lot in order to get to that transition state. Just to illustrate the importance of proximity and orientation for reactions, I want to show you some of the different reaction rates comparing molecules that are free versus molecules that are placed in closer proximity or in a better orientation. So if we compare two different molecules that are going to react, so we have a nucleophilic acyl substitution reaction here between a carboxylic acid and an ester. If you just have these two free in solution, they can react together to create an anhydride. But this reaction is going to be relatively slow, right? They have to find each other, they have to get in the right orientation, make our reaction. Somewhat of a slow process because of the entropy associated with that. If we link these together, so we put them on opposite ends of the same molecule, now it's much easier for them to find each other, right? They're linked together on the same molecule. But they still have to rotate into the correct orientation for that reaction to take place. So you'll notice our rate has increased by 100,000 times. Now, if we further change our structure so that not only are they linked on the same structure, but they're also locked in the correct orientation. So we put it in this ring structure here so that they can't just freely rotate however they want. And so they're both kind of stuck sitting right next to each other. Now it's much easier for them to find each other, and we have a 100 million times increase over our original rate. So again, this is essentially what our enzyme is doing. It's just grabbing up those two structures, putting them close together in the right orientation, and allowing our reaction to speed up. The second mechanism that our enzymes use to lower that activation energy barrier is they bind to our transition state better than the product or the substrate. So this idea was first suggested by Linus Pauling, who was a famous biochemist. He won the Nobel Prize for biochemistry. So he noted that enzyme active sites are complementary to the transition state of the reaction. And again, they bind it better than their substrates, and they use that additional binding energy to help, again, to lower that activation energy barrier. This is a largely delta H-based effect of just using enthalpy of binding to help to lower that free energy of the transition state. Now, to understand why it's so important to bind the transition state specifically, it's helpful to use an illustration here. So imagine that we have an enzyme that's going to bind to a metal stick, and our goal is to break it. So for that metal stick reacting, you can imagine this sort of reaction here, where we have a transition state where it's bending, and you got a lot of stress on the stick, and then your product's where it's broken in half. Right? So without the enzyme present, we get this sort of graph. Our substrate, we go through this high activation energy barrier as we're bending it, and then it snaps to create our products. So now let's bring in an enzyme. And of course, we want to form a strong interaction to our substrate, right? So we have, in this case, we're using magnets to surround our metal stick, right? So we can bind it very tightly in our substrate, or in our enzyme substrate complex, rather. Now, if you look at the free energy, right? So when we bind to our substrate, notice you have this very large negative delta G, right? That's because of all those strong interactions that we're forming to our stick here. But if we then try to bend this substrate to try to break it, it's actually going to be harder to bend it, right? Because now we have our magnets that really want to stay bound to that straight stick here, right? If we bend it, we would actually have to break some of our interactions here to that substrate. So if you look at the free energy diagram, we actually haven't done anything to help our activation energy here. Yes, we made a very strong interaction with our substrate, but it doesn't really help us. In fact, it actually hurts us because now we have to break those same interactions as we move into our transition state. And so our activation energy barrier gets higher. So this sort of enzyme that's perfectly complementary to our substrate would actually slow down the reaction rate. it would essentially just bind your substrate and hold it in that original reactant structure and not allow it to react. So instead, we want to change our enzyme such that it has a binding site that will still interact with our substrate, right? So we get these interactions here at the poles of our little stick here. But it increases the interaction as we switch to our transition state, right? So when our stick bends, it gets more interactions here with the magnets up here, right? So our enzyme substrate complex actually has this bent shape that essentially pulls our substrate into that bent shape of the transition state. Then we're able to break it to our product and release that product at the end. So notice the change in our graph here, right? We have our substrate, and then we bind the substrate in our enzyme, and you'll notice it's a relatively small difference here in terms of delta G, right? We don't have super high affinity for the substrate, but instead, we have very high affinity for the transition state, and we are able to create this lowering of that activation energy barrier through the delta H contributed from this extra interaction here. So now our activation energy barrier is much smaller, which again will lead to a faster reaction. So again, if you bind the substrate too tightly, you'll actually slow down the reaction because it won't want to change into the transition state if the transition state isn't going to bind more tightly to your enzyme. Now that we've seen the general thermodynamic approach of our enzymes, I want to look specifically at the general catalytic mechanisms that we commonly see for our enzymes. So we'll generally see our enzymes use one or more of the following different mechanisms. So we'd have an acid-base catalyst that has acidic or basic groups on the protein that will then be giving and taking protons from our substrate. So of course, this is very familiar again from organic chemistry, where we're often adding strong acids or strong bases in order to encourage a particular reaction. Only in this case, of course, we're using a much more controlled and, of course, weaker acid or base that's just reacting with our specific substrate. We can also have a covalent catalyst, where we're actually changing the reaction path by reacting between our enzyme and our substrate and forming a covalent bond. And then we're reacting again and eliminating that covalent bond to restore our enzyme to its original structure and to then create our product. So our enzyme actually temporarily participates in the reaction, links itself to the substrate in a covalent bond. We also have a wide variety of metal ion catalysts. So these can be used to shift pKa values, right? Again, that's what we talked about earlier, how charges will affect pKas and change the ionization of, that could be the substrate, or it could be amino acids within your enzyme that you're using for acid-base catalysis. Metal ions can also be used to help with redox reactions, whether they're transporting electrons from the substrate to a product, or they could also be used to help to stabilize charge in our transition state as we're going to our product. Right, so there's a variety of different ways that we could use our metal ion catalyst as well. Most of our enzymes will use some combination of these three kind of general mechanisms. So here's an example of what a general acid-base reaction might look like. So here we have our reactant species. So we have an alcohol that's attacking an amide to do a nucleophilic acyl substitution, right? So our alcohol is replacing the amine to form an ester. So without catalysts, right, we react our alcohol and we end up with this charged intermediate that then is actually more likely to just break apart and reform our reactants rather than reacting to form our products. So what our enzyme needs to do is help to stabilize that reactant, right? So what we're going to do, or help stabilize that intermediate form specifically. So our enzyme will transfer protons from from itself to our reactant in order to eliminate those charges and stabilize our intermediate. So here we want to take off the hydrogen to help stabilize this oxygen here. And then we want to add our hydrogen over to our nitrogen to make that more destabilized to then allow it to break off to form our products. So of course, this process can occur normally just with our intermediate reacting with water or with a base or acid catalyst. But within our enzyme, we're going to use specific proton donors or acceptors, like usually our acidic or basic amino acids, to help to push this process along and control it so we get our specific products formed. So as I mentioned, our acid-base catalyst will be most commonly used in this acid-base catalyst mechanism. So our glutamate and aspartate with our carboxylic acid. So it could act as a proton donor in its protonated form, or it could act as a proton acceptor in its deprotonated carboxylate form. Lysine and arginine, of course, have our amine group that can act as a proton donor if it's positively charged, or a proton acceptor if it's neutral. Cysteines actually also can be used. So you might remember that thiols are slightly more acidic than alcohols because of the weaker bond between our sulfur and a hydrogen. They could be used as acids or bases depending on the protonation state. Histine, of course, also has a nitrogen that can be used as a acid-base donor or acceptor. Histine is nice because it has a much lower pKa, so it's more likely to be able to switch our donate or accept protons, since it's kind of right around that natural state where it can be easily protonated or deprotonated. We can also use our alcohol-based functional groups, so serines or threonines or tyrosines. These, of course, are a little bit harder to use because they do have much higher pKa's, but we will still see them used as, again, proton donors or acceptors, depending on the scenario. One specific example of an acid-base catalyst is our Phosphohexose Isomerase enzyme. So this enzyme is part of our glucose breakdown process to turn glucose into energy. So we take our glucose in the form of glucose 6-phosphate, and we use a base here in the form of glutamate. So we have our deprotonated glutamate that takes a hydrogen from our glucose in order to shift its structure. So we are tautomerizing our aldehyde here from the aldehyde form to our enol form. We accept our proton from our glutamate. We then take the proton back from our glutamate, and we rearrange our molecule to the ketone form. Right? So we essentially switch back and forth from our aldehyde to an enol to a ketone as we transfer that proton back and forth from the glutamate here in our enzyme. So the next mechanism that we'll look at is our covalent catalyst mechanism. So this is where we're forming a transient covalent bond between the enzyme and substrate. Now, of course, normally we don't refer to covalent bonds as being transient, but in this case, of course, we're going to react and form that bond, and then immediately react again and break that bond. So even though it's a strong bond, it's still only there temporarily between our enzyme and substrate. So again, we're essentially changing the reaction pathway. Uncatalyzed, we would just be breaking our substrate apart to form our products here. But then with the enzyme, we're breaking it, but we're bonding part of it to our enzyme. So here we have a bond forming between product A and our enzyme, and then we release that to form our free products in our enzyme. So of course, one of the requirements for this sort of reaction is that we're going to need a nucleophilic site on the enzyme. So we've seen the different nucleophiles on our amino acids before. Some of the most commonly used nucleophiles are serine, the thiolate from a deprotonated cysteine, We have, of course, our amine groups on things like lysine or arginine or our asparagine and glutamine. We can also use our carboxylates from our glutamate or aspartate, things like that as well. So in this sort of covalent catalyst mechanism, we're going to be using our nucleophile to attack our substrate to form a bond there. So it's essentially going to be a nucleophilic substitution. So in our glyceraldehyde 3-phosphate dehydrogenase, again, another enzyme that's critical for breaking down glucose, our substrate comes in, our glyceraldehyde 3-phosphate, and we have a thiolate here from our cysteine that attacks our aldehyde here in a nucleophilic reaction. So we attack, link on, and then we're actually going to react with this, we call this enzyme cofactor over here. It's another molecule that's bound within our enzyme that's helping to catalyze this reaction. And this is then going to react, and it's going to take a proton from our substrate. So that's kind of like a acid-base catalyst mechanism in some ways. And then we now have our substrate linked covalently in our enzyme. We then bring in another nucleophile in the form of phosphate to react through a nucleophilic substitution reaction and break that bond between our enzyme and our substrate. And this then releases our final product. The last general mechanism of catalyst is our metal ion catalyst. So again, this is going to involve a metal ion bound within our enzyme. And we'll use that to facilitate binding through stabilizing negative charges. Again, it can participate in oxidation reactions, as I discussed previously. So the example here is our alcohol dehydrogenase enzyme. So this enzyme can be used in things like yeast to produce alcohol, produce ethanol. In humans, we can also use it to break down ethanol that we might consume. So what our enzyme does, it uses a zinc bound within our enzyme to associate with our negatively charged oxygen intermediate, right? So we do a nucleophilic attack on our reactant, our substrate here, which will result in a negative charge on our oxygen after that nucleophilic attack. Our zinc here just helps to stabilize that transition state in order to lower that activation energy barrier. So this is a relatively simple use of that metal ion catalyst. Again, there are other more complicated uses where we could have it participating in some sort of redox reaction, helping to transfer electrons. But yeah, this is just one of many different ways of using that metal ion catalyst. So in this lecture, we've learned some of the general different mechanisms of our enzymes. So they'll use these, again, to lower that activation energy barrier and speed up our reactions to a biologically useful time frame. In the next lecture, we're going to be looking at how we can calculate what the rates of our reactions are from our enzymes, and then we'll be using that to then look at how we can regulate our enzymes as well in order to precisely control our biological reactions.